Hello, my dear students. In this class, let us discuss about the bacteriology, the ultrastructure of bacteria. We have in the previous class we have discussed about the cell wall, the peptidoglycan layer, the structure of peptidoglycan layer, etc. Apart from that peptidoglycan layer, there are other components which plays a very important role in the bacterial cell. So this is an anatomy of ultrastructure of bacterial cell. You can see the capsule, which is the completely outer covering of the bacterial cell, followed by the cell wall. You can see the cell wall here, light orange color, followed by plasma membrane. You can see mesosomes, cytoplasm, nucleoid, which is nothing but the genetic material, flagella for the movement, plasmids, inclusion bodies. Inclusion bodies plays a very important role in the bacteria to survive under different conditions. Polyribosomes, there are different types of ribosomes present in the cell of the bacteria, and you can see the pilus or pili, which helps in the movement and attachment of bacteria to any surface. So this is an remembrance how to remember the components of the bacteria. First comes the capsules, followed by the cell wall, then comes the cytoplasm. So there are three different main components present in the bacterial cell. First component, let us discuss what is capsule. Capsule here, it's a gelatinous substance or which is composed of polysaccharides or polypeptides, sometimes both. The capsule, the first thing is the covering of the bacteria. The second thing is it helps the bacteria from desiccation. Just imagine if there is no nutrient around, then the cell may get desiccated. It prevents from desiccation. At higher temperature or during drying, the environment may change. That time the capsule prevents from desiccation. The back Just imagine if you are using antibiotic to treat the bacteria. That time, if capsule is there, the capsule will prevent the antibiotic action against the bacteria. Along with this, due to the gelatinous substance, it helps the bacteria to stick to the surfaces like plant roots, human teeth, tissues, or any kind of substances due to its sticky nature. Right? So let us move on to the cell wall, which we have already discussed that this cell wall provides the rigidity, provides the strength, provides the mechanical support for the bacteria, which is composed of peptidoglycan layer, or this peptidoglycan layer is made up of NAG and NAM. NAG means N-acetyl glucosamine and NAM is N-acetyluramic acid. Along with this, you have four amino acids which is linked with the tetrapeptide, right? So this we had discussed in our previous class. Now moving on to the role of plasma membrane. Here, my dear students, as the bacteria doesn't have mitochondria, so in order to carry out protein, in order to carry out the metabolic processes, they require various enzymes. These enzymes are active, and the site of uh, metabolic activity takes place in the plasma membrane. The plasma membrane is made up of lipoprotein. My dear students, the plasma membrane is made up of lipoprotein, and it controls, it's like a barrier which controls the exit and the entry of the molecules and ion. It's like a gate which prevents the entry of unwanted substance. It allows only the nutrients or molecules, ions, which are essential for the bacteria for its growth and multiplication. Along with this, it's also add the structural support to the cell, and it is a site where major enzyme activity takes place, especially The respiratory chain reaction takes place as there is no mitochondria. All the functions of mitochondria in the prokaryotic cell is carried at the site of plasma membrane. You need to remember this. And there are certain bacteria which are having photosystem, which means they are phototrophic bacteria which converts the light energy. They depend on the light energy or light source. During such bacteria are also having photosystem in the plasma membrane. So let us see how this plasma membrane looks. Here you can see the lipophospholipid. The phospholipid is structured and forms the plasma membrane. Within the plasma membrane, you have integral protein. You have protein channel. This channel allows the transport of proteins to the cell. Along with this, you have carbohydrates and you have surface proteins and you have filament or cytoskeleton which provides the strength or the support for the cell. You have peripheral protein here, you have glycoprotein. See, here in plasma membrane, you need to remember that it is made up of phospholipid layer, and here the phospholipid layer is embedded. Within the phospholipid layer, you have different types of protein such as glycoprotein, number one, peripheral protein, number two, and they have protein channel to transport protein. They have globular protein and they have integral protein. Along with this, they have surface protein as well. They also have filaments or cytoskeletons. This cytoskeleton provides the strength for the cell. What are the functions? One first function we came to know that it allows the various metabolic activities. The best example is respiratory chain reaction takes place in plasma membrane. The second thing is it acts like a barrier to prevent the entry and exit of the molecules. And now let us see what are the other functions of plasma membrane. Here you can see the plasma membrane has selective permeable membrane, meaning allows certain molecules can pass through them. Water, oxygen, carbon dioxide can easily travel through the membrane. Generally, ions such as sodium, potassium, and polar molecules cannot pass through this membrane. They must go through specific channels or pores in the membrane instead of freely diffusing, which means the water, oxygen, and carbon dioxide can easily penetrate through this membrane, whereas the generalized ions such as sodium, potassium need to pass through a particular channels. There are different channels here. You can see the protein channel. Similarly, you have different channels which allows the movement of ions. Also, sometimes what happens, the larger molecule is consumed by the cell. For example, if we eat much and we feel that we are completely heavy, we need to digest it. Similar way, the bacteria also consumes a larger molecule or larger content of ions through different channels. During that, the bacteria should break down these molecules, and the process is carried out through endocytosis. Endocytosis is a process where larger molecule is consumed by the bacteria, and within the bacteria, this larger molecule is broken down into a smaller molecule and it is utilized by the cell. Similarly, exocytosis is a process where the bacteria is, for example, the bacteria is living in the water and there is a larger molecule which is required for the bacteria, but the size is big. What happens during that time? The bacteria secretes some enzymes, secretes some, release some metabolites so that uh, larger molecule is broken down into a smaller molecule so that it can easily pass through this uh, plasma membrane, right? Along with this, the plasma membrane also carries out cell signaling. they carry out cell signaling. There are various markers present in the plasma membrane which helps in identification of bacteria. So one bacteria try to communicate with the other bacteria using cell signaling. This cell signaling process also takes place in the plasma membrane. So majority of part of the plasma membrane is made up of phospholipids. Let us see the structure of phospholipid. The phospholipid is having hydrophilic head and hydrophobic tail. The hydrophilic head is nothing but a water-loving molecule, and the hydrophobic tail is water-hating molecule. Here you can see this phospholipid is attached together parallelly to form plasma membrane. Within this, you can see the protein channel, globular protein, surface protein, etc. But in order to form this, phospholipids are arranged in a sequential manner to form a plasma membrane. So moving on to the proteins, proteins are fixed between the lipids that make up the membrane and allows certain molecules to pass through it. As we have seen here, even in this pictorial diagram, you can see protein channel which allows the passage or entry and exit of the various molecules. You have membrane protein which we have discussed, globular protein, carbohydrates. The plasma membrane is also having carbohydrates. These carbohydrates are essential component of plasma membrane, and carbohydrates in the membrane are part of glycoprotein, which we had already seen in the structure, the glycoprotein, which are formed when carbohydrate attaches to the protein. The carbohydrate and protein forms together to form a glycoprotein. Glycoprotein play an important role in the interaction between cell. It helps in cell addition, which is nothing but attachment of cell. They try to attach the cells try to attach each other with the help of glycoprotein. So moving on to, like, first we completed the plasma membrane, first we complete capsule, then plasma membrane, and now we will go to the most important component of bacterial cell, which is nothing but cytoplasm. The cytoplasm is a semi-transparent uh, liquid. It is a thick, it contains all the inclusive bodies along with ribosomes, nucleoids, plasmids, etc. So here you can see the ribosomes are present, nucleoids are present, plasmids are present, cytoplasmic inclusion bodies are present, spores and cysts are also present in the cytoplasm. Spores are nothing but the formation of a structural body which, upon getting the favorable conditions, starts multiplying into a new living entity. So these spores helps in reproduction and multiplication. Composition of, let us move on to the composition of plasma cytoplasm. The cytoplasm contains 80% of water, which is nothing but liquid, and this liquid component in the cytoplasm is called as cytosol. Within the cytoplasm you can see nucleic acids, DNA, RNA, enzyme, amino acids, carbohydrates, lipid, inorganic ions, and other functional entities. The cytoplasm is also a site for majority of microbial bacterial metabolism. Here within the cytoplasm, there are various bacterial metabolism takes place, which is essential for the growth and multiplication. Along with this, bacterial cytoplasm also contains helical actin-like proteins that, along with the cell wall, contributes to the shape of the bacteria. Ribosomes, as we have already studied, ribosomes play a very important role in protein synthesis. which is required for the growth and multiplication of every living organism. Here in the bacteria, you have two subunits, 30s subunit and 50s subunit. Let us see what are the composition and role of ribosomes in the prokaryotic bacteria. Ribosomes in prokaryotes exist as inclusion bodies within the cytoplasmic matrix. It is present within the cytoplasm, which appears as granules. It will be in the granule form, dense and complex structure made up of RNA and protein. It holds 30% of protein and 65% of ribosomal RNA. Ribosomes are universal membraneless organelles that are common in all groups of living organisms, as I discussed now. The prokaryotic ribosomes are dense structure which solely contains RNA and protein. And here, the ribosomes in the prokaryote cell are thoroughly distributed. Throughout the cytoplasm, it is distributed. There are two subunits, 50s and 30s. The functions of individual subunits will be discussing here. 30s subunit, it provides the binding site for the incoming transcribed mRNA. Also, the smaller subunit of prokaryotic ribosome ensures specific base pairing between the codons and anticodons of mRNA and tRNA respectively. So this 30s subunit helps in the attachment of transcribed mRNA, right? And it helps in the binding of or pairing of codons and anticodons of mRNA and tRNA. Here you can see the mRNA and the tRNA. The codons and anticodons helps in formation of amino acids, and this amino acids grow as a polypeptide chain. The 50s subunit is comparatively larger than the 30s subunit, which mediates the peptide bond formation during peptidal transfer reaction. It also acts as a site of inhibition for many antibiotics, including macrolides, chloramphenicol, etc. So here you can see the 50s subunit of prokaryotic bacteria, which helps in growing of polypeptide chain. This play a very important role in growing of polypeptide chain. It also helps in protein folding after the translation is done. Next component in the cytoplasm of the bacteria is a nucleoid. Nucleoid is nothing but a chromatin body. It contains the chromosomes. The bacterial nucleoid has no nuclear membrane or nuclei. It is diffused in the center of the cytoplasm. The chromosomes of bacteria is single long haploid molecule of double-stranded helical supercoiled DNA. In most bacteria, the two ends of the double-stranded DNA covalently bound together to form a closed circle, which means one end of the DNA and the other end of the DNA are closely connected with covalent bond to form a closed circle or closed structure. The chromosome is approximately 1000 micrometer long and frequently contains as many as 3500 genes. And here plasmids, which we have already discussed, what are plasmid, what are the different types of plasmid, what is the role of plasmid in our previous lecture. Here I will give you an overview. It is self-replicating body. It carries certain special genes which are responsible to carry out a specific function of the bacteria. It doesn't participate in the reproduction part of the bacteria, but they carry additional genetic information and they can replicate independently. Plasmids usually contains 5 to 100 genes depending upon the type of plasmids. Transposons are nothing but a jumping. Transposons are nothing but jumping genes. They are also called as jumping genes, mobile genetic material elements, where within the chromosomes or within the plasmids, these jumping genes try to move from one location to another location. And this transferring is due to the presence of transposase enzyme, which helps in movement of the jumping genes from one location to the another location. Inclusion bodies is like a reservoir of cytoplasm of in the cytoplasm reservoir in the cytoplasm where they reserve certain foods for the bacteria in order to so that it can utilize when there is non-availability of nutrients or non-availability of uh, elements, then these inclusion granules or inclusion body play an important role for the bacteria to survive. It is like storing food in the refrigerator. Whenever you want, you will take, you will consume that food. So let us see what are the different types of inclusion bodies. We have glycogen. Glycogen, many bacteria, example E. coli, the composition made of the composition of glycogen is polyglucose, and this reserves the carbon and energy source. When there is no carbon source available to the bacteria, then this glycogen will supplement the carbon which is reserved inside the glycogen. PHB, polybeta hydroxy butyric acid. Many bacteria, example Pseudomonas, it also reserves the carbon and energy resource. When there is exhaustion of carbon source, then this PHB releases the carbon, provides the carbon to the bacteria. Polyphosphate, it is also called as volatile granules. This can be seen in many bacteria. Best example is Corynebacterium, and it reserves the phosphate and which is used the reserve phosphate, possibly a reserve of high phosphate energy which is used during respiratory chain reaction and other enzymatic reaction. Sulfur globules. This is especially found in specialized bacteria such as phototrophic bacteria, sulfur bacteria, etc. It reserves the electron so that it can be utilized during the photosynthesis reaction. Gas vesicles, especially these are found in aquatic bacteria, and it is made up of protein, and here it provides flotation in the vertical water column. The function is flotation. Next is parasporal crystal, especially found in endospore forming bacteria. There are various bacteria which forms endospore within the body, within inside the cell. You can see the endospore formation, and the function is yet to be elucidated or yet to be known, but it is toxic to certain insect or certain living organisms. Magnetosomes, even this is found in aquatic membrane, aquatic bacteria, and it helps in orienting and migrating along geomagnetic field lines, moving around, moving according to the geomagnetic field itself, magnetosomes. Next is carboxysome. Many autotrophic bacteria here uses carboxysome for the fixation of carbon dioxide. So these were some of the commonly found granules present inside the cell as inclusion bodies. In order to control the bacterial growth and replication, there are various antibiotics which are used. And how this antibiotic work? For example, if you take penicillin, The penicillin inhibits the cell wall synthesis. When there is no cell wall synthesis, then there is no formation of cell. Ampicillin inhibits the cell wall synthesis. Similarly, along with cell wall synthesis, there are protein synthesis. There are antibiotics which controls the protein synthesis. Example, tetracycline, streptomycin, etc. At the same time, you have rifampin and quinolines which inhibit RNA and DNA synthesis. So these antibiotics target differently such as cell wall synthesis, protein synthesis, RNA synthesis, cell metabolism. They have a different targets. So with this, let me conclude today's session which gives an overview of the components of cell. Thank you for your patience. I wish you all very best in your upcoming examination. Good luck.